Thank you very much, Siddharth, for agreeing to give a Build a Cell seminar, and you can get started.
Awesome. Well, hi everybody, and thanks for joining. Thank you, Kate, not just for inviting me here, but also keeping this beautiful tradition of Build a Cell seminars so active over so many years. It's quite incredible. So my name is Siddharth Deshpande, and I actually gave a seminar back in 2020. I distinctly remember it was the last seminar during COVID, and I had just started my lab back then. And now so many things have happened since. So I'm really excited to give the updates with this second edition of the seminar for me. So maybe a little bit about where we are situated. So we belong to this beautiful flatland, European flatland called the Netherlands, and in particular, my lab is basically situated in Wageningen University and the Department of Physical Chemistry and Soft Matter. And the department itself is very diverse. So we do all sorts of stuff, right from polymer materials and coatings to, studying flow of complex fluids, and also do a lot of soft matter instrumentation. My interest definitely lies more in the self-assembly of biomolecules. And if I look at my lab, that is also pretty diverse. So we use a lot of microscopy, spectroscopy, but also physical chemistry and microfluidic tools to study basically biological soft matter. And our current interests are ranging from protein condensates to membranes vesicles, but also we are going a little bit into biosensing. So this is sort of the visual overview of what we are currently doing in the lab. So we are very much interested in protein phase transitions of synthetic as well as natural proteins. And this will come a lot in this talk later on. We are also looking at stability of Pickering emulsions. So normally, instead of you normally have the surfactant stabilized emulsions, but you can also have particle stabilized emulsions, and then they are called Pickering emulsions. We are looking into that. We are looking at actually emulsion droplets as biosensors. So what you see here are liquid crystals that are acting as biosensors and changing color in response to amplifiers. We also use emulsions to actually look at on-chip directed evolution, something that we have been doing recently. For a long time, my interest lies in this minimal immersion systems, and which is very much connected to the building synthetic cells. How can a small set of molecules give rise to immersion properties, something that is unexpected? And that, of course, is tightly related to building synthetic cells, playing Lego with biomolecules, basically. And this is what I'm going to talk about. So in a sense, as a community, what we want to do is, build life from scratch, let's say, and starting with individual biomolecules, start increasing the complexity and see if we can get lifelike materials. At the same time, for me, it's interesting to look the other way. How simple can we go and still maintain some sort of immersion properties at this lowest minimal level? So this is also something that we are working on. So what I'm going to talk today is about compartmentalizing synthetic cells. So how can we compartmentalize our confinement? Because I think this is a very important point if you want to keep on building more and more complex synthetic cells. If you look at living cells, it's an organized chaos in there. There are lots of different structures inside the cell, both membranous, but also in the last 15 years, we have been realizing that there are lots of membraneless organelles. And this phenomena behind this formation of membraneless organelles is generally known as liquid-liquid phase separation. Now we know a lot of membraneless organelles, nucleolus, P granules, pyrenoids, etc. So can we use this sort of phase separation phenomena also for building synthetic cells? Broadly speaking, phase separation can be divided into two sorts of systems. One is a segregative phase separation, where the polymers basically form two different polymer-rich phases. For one phase made up of polymer 1 and the other one made up of polymer 2. There is also the associative phase separation, where the two components come together to form a dense phase, which is in equilibrium with the dilute phase. Now, in the context of biology, these are basically multivalent biopolymers. So not necessarily purely a polymer, because, for example, ATP and polylysine can also bring about associative phase separation. Generally, how you look at these phase separation systems are through phase diagrams like these. So for SPS, so the segregative phase separation, you basically have concentrations of these two different polymers. And if you have high enough concentration, they go from a homogeneous state to a phase separated system. Similarly, for the associative phase separation, you typically look at the concentration again, but also as a function of a trigger, for example, pH, temperature. And there as well, from a one-phase region, you end up into a two-phase region. Now, we are going to use both these systems, as you will see as in the next slides, basically. But let's also talk a little bit about the vesicle itself that we want to complexify, let's say. So in general, if you look in the literature, people use three different kinds of vesicles, membranous vesicles. You can start with a single emulsion, which is a very simple system in a sense. It's a water in oil emulsion stabilized by surfactants. On the other hand, you can have a liposome, which is basically the best mimic of a living cell, best minimal mimic, because the boundary is made up of a lipid bilayer. But there is also something very interesting in between, and this is known as a double emulsion. And what it is basically is it's a water in oil in water emulsion. So the boundary is not necessarily made up of a lipid bilayer, but rather a thin layer of oil surrounded, again stabilized by surfactants. And in general, as you go from left to right, the biomimetic nature increases, but also the production difficulty in general increases. Now, how do you produce them? Well, of course, there are lots of different methods to produce these vesicles, but we very much like to use microfluidics to produce them. For simple reasons, it's cheap. There is a rapid prototyping to make such devices. I mean, I'm showing you a typical lab-on-a-chip device here. You can actually see small channels engraved into this PDMS through which we can flow solutions. We can use very low volumes. I mean, I say 100 microliter here, but we can even go lower. And it's a high throughput system. And the reason microfluidic works, or it's different from a bulk process, is I think can be nicely seen here, because the turbulent flow that you normally see in a macro system is absent, virtually absent at the micro scale, where there is purely a laminar flow, and the only mixing that happens is via diffusion. And now you can really make use of this this phenomena to make interesting structures. And one of the things that I developed during my postdoc is OLA. So that's octanon assisted liposome assembly. It's a on-chip technique to make liposomes. And you can again see nicely how we can blow these bubbles. We have a very effective bilayer formation. We can encapsulate proteins inside these vesicles. So these vesicles we can collect, and you can basically see a nice fluorescent image again of these vesicles. Most of them look pretty nice. Of course, there are some disturbances here and there, but ultimately this is soft matter. You cannot get picture perfect population. All right. So I think this is enough introduction about the different tools that we are using. And now the question I'm asking is, how can we make compartments inside vesicles, but not just compartments? How can we trigger them reversible? Because just like in cells, I mean, not all these organelles are present at all the times. They get triggered by different stimuli, and we would like to do the same with our compartments. So now if you think about the fundamental triggers that that a biological system experiences, I can think of 4. So that's pH, a very fundamental biochemical trigger, temperature, salts, ionic strength basically, and the concentration of the solutes itself. So can we think of a system that can that is responsive to all these stimuli? And the answer is yes. So we can design multi-responsive peptides to that respond to all these triggers actually. So and this is in collaboration with Renko de Vries lab, which is basically next doors, and they have been doing this beautiful work with elastin-like polypeptides, so ELPs in short. So these are again synthetic mimics of the natural protein elastin. They have a very particular sequence. It's basically GXGVP, and that is repeated n times. G is glycine here, valine, proline, and X is the guest residue. So what we designed in the last 2 years are basically pH responsive ELPs. So normally ELPs are temperature responsive, but we also made them pH responsive. So basically they respond to all 4 now: pH, temperature, salt, and of course the protein concentration itself. And here is, for example, a series of pre's that we developed, and I'm highlighting the pH 46 and pH 36 because these are the 2 proteins that we are going to use basically in the latter experiments. So as you can see, each of these pre's have a distinct pH transition at 21 degrees, but also that transition is much higher at 37 degrees. And you can nicely see how tunable basically these protein sequences are based on which sort of hydrophobic amino acid is there. We can tune the pH transition. And you can see a nice example where between pH of 6 basically and 5.8, you suddenly get condensation of, in this case, this pre-H46. All right, so now we are going to use these pre's. And remember the double emulsions that I was talking about? Instead of liposomes, here we are using double emulsions. Because actually, if you do not care too much about the membrane interactions, double emulsions are an equally effective platform to study confined systems. And actually, they are easier to produce. So here you see a double emulsion, a typical double emulsion production. It is the inner aqueous phase along with the oil phase that gets pinched off at the outer aqueous and forms these double emulsions. It is the encapsulated proteins. And again, you already see it in the video, but if you just collect them later on, they are nice and homogeneous, monodisperse. You can control the size. Here they are about 30 micrometer in diameter. All right. So what we are doing basically is encapsulating our pre's inside. And I'm going to show how different triggers can lead to this membranous organelle formation inside our vesicles. So here is again a straightforward experiment. We produce the double emulsions, collect them, and now we subject them to basically a pH trigger. Here we are using very drastic pHs just to also show how stable the double emulsion system is across a range of pH 1 to 13. So when we trigger basically with pH 1, you get this beautiful formation of the membranous organelles inside our double emulsions. And when we again put a basic trigger, it goes back. Here is kind of a time lapse of this condensation process happening. You can see the coarsening. You can see the first individual coacervates happening, forming, and then they fuse over time to form a single coacervate. You might take a look at the time and say it's slow, and it is, and that's because the double emulsion has a thin oil boundary, and it takes time for the pH to equilibrate across this boundary. We can also do some simple analysis. We are basically just simply looking at the intensity of the standard deviation as the membranous organelle forms. The intensity the standard deviation shoots up, and then it again goes down. If you look at the efficiency, it's not the forming efficiency is very good, but they do not dissolve. They do not all dissolve. So some population still remains. And actually, we think it's because of the long time that is required for this dissolution. And in that time, some evaporative effects come into play. The concentration of proteins increases, and that again promotes condensation. What about temperature? So again, a simple experiment: harvest the teas and trigger it to like 46 degrees. And we could actually make even 2 cycles of nice organelle formation quite quickly, and again dissolution. But at the second cycle, you see a little bit of kind of non-dissolution, and that again we think is because since we are heating things, water evaporates and maybe some osmotic effects come into play. So we were thinking that's all good, but can we also use another trigger? And this is in bulk. And what we see basically is if we do the experiment and just leave the double emulsion suspension to evaporate, you again get this beautiful membranous organelle formation inside each of these vesicles. And that's simply because over time, the water is evaporating outside. So there is a hyperosmotic shock, and the double emulsion size decreases, and you get again basically a trigger because of the ELP concentration increases and also the salts. So we want to do this on chip. Wouldn't it be wonderful if we trap our double emulsions and subject them to very controlled triggers repeatedly, and to do this on-off formation? So we made traps. We trap our double emulsions. The trapping efficiency could be perhaps improved, but this is also very good for now because what we want to do is we want to focus more on this triggering. So what we are doing is actually making things a little bit more sophisticated. So we use what we call a DAW junction. So it's a dial-a-wave junction, and it works the following way. So the trap array is in this middle channel, and then you have 2 inlets for 2 different triggers. And you can imagine when the inlet 1 has a higher pressure, that fluorescent dye will go into the trap array, and that will be the trigger. If you increase the inlet pressure on inlet 2, that will push away the inlet 1, the fluid from inlet 1, and you can basically tune the trigger. You can see it much better here in the video. You can see that as I increase the pressure on inlet 1, you can see the fluorescence going into the traps. And if I increase the pressure on inlet 2, it will go back again. So, yeah. So this is how the entire microfluidic chip looks like. We have the 2 triggers. For example, it can be acidic buffer, basic buffer. Here is the flow control unit, some waste channels, and here are our double emulsion traps. So I want to simply show you a movie where and this is taken over hours, and I will play it, and you can nicely see as I switch on the trigger, you will start getting this formation of the membranous organelle inside. And now when Chang switches it off, the organelle dissolves very quickly. And then he switches it on again. Again, you have the formation, switch off, it dissolves. You can basically do it till you get bored. So I think here Chang did it 7 times. And at different time intervals as well. So fast, or sometimes just with a long time gap between them. It doesn't matter. Every time you switch on the trigger, you get this beautiful membranous organelle formation. So that works very nicely. And here is a time lapse basically showing how we can nicely tune the membranous organelle formation and dissolution. So this was kind of the first story I wanted to share, which is also published, that we have taken ELPs and double emulsions as a model system to bring about condensation inside synthetic cells. And I think it's a very effective platform. The ELPs themselves, you can, based on the sequence, you can really engineer it to how you want it. The double emulsions itself are very, very robust vesicle systems to play with. And as long as you don't need a membrane interaction, they are perfect to work with. Okay, but now let's make things a little bit more complicated. So I talked about right now the condensates, but I also previously showed the SPS, right? So the segregative phase separation. And we were thinking, can we bring both in play inside liposomes? So if you look in literature, people have worked a lot with liposomes and associative phase separation, liposomes and segregative phase separation, or the two types of phase separations. But all together is, yeah, you almost don't see an example of that. And that's what I will be talking about now. The story actually just got published in ACS Nano last week, so right in time for this talk. So let's start with it. So as a model system for this segregative phase separation, we are using a classic system of polyethylene glycol, PEG in short, and dextran dex in short. And what we did first was characterize our PEG dex system. And we basically looked at the phase diagrams at different conditions. So different pH from 3 to 10, different variations of buffer, different viscosity, glycerol, no glycerol. And what we see is the PEG dex itself is a very, very robust system. By the way, this is in collaboration with Atul Parik's lab at UC Davis. A very nice collaboration. We really enjoyed it a lot. So this is a cumulative phase diagram, basically, of all of these things, showing that it's a very robust system. And we can really change a lot of parameters, but we will always get the phase separation of PEG and DEXT within these concentrations. And for the associative phase separation, now we don't use ELPs here, but we use another classic model system of polylysine, PLL in short, and ATP. And what is interesting is that the polylysine shows differential partitioning. So polylysine really likes to go into the DEX-rich phase and not into the PEG-rich phase. But the ATP doesn't really care. So ATP just goes equally in both the phases. So that gives it an interesting interplay of these components. So now let's have first some bulk studies. So if we simply put all these four components together and trigger phase separation, what we get is already quite interesting. We get, if you look in the bright field, we basically get dextran droplets in PEG, and within the dextran droplets, we get our polylysine ATP coacervate. The dextran also actually partitions further into coacervate. This is a kind of an unwanted side, unwanted outcome, but it doesn't really matter. It's just the fluorescence dex that is partitioning further. So you can actually ignore that a bit. You can actually see how physically confined they are. So if you put a little bit of flow over the cover slide, you can still see that these coacervates still remain confined within the dextran droplets, basically. So they're really, it's physically confined. But not only that, if you do FRAP, and if you, for example, for a non-confined system, if you bleach, of course, you expect recovery, and that's what you get. You can also see it here. But in an SMC, so I'm calling these SMCs for SPS-based membraneless confinements now. Sorry for another short form. If you do it in SMCs, you actually do not get any recovery. And that's because the dilute phase is really, or the SMC is really isolating these coacervates from the surrounding environment. And we think it's a very interesting environment then presented to the condensate, that it remains physically and chemically isolated. So as shown in this sketch, the SPS is really controlling how the APS should behave. So now let's start with some fun experiments of actually encapsulating all the four components now. So we did this to start with, we did it in double emulsions because just to show what happens in a non-membrane interaction. When we do it, what you actually get is a multi-phase structure as you would have expected based on the bulk experiments, right? So here is the double emulsion, and then you have your dextran and the dextran-rich phase, and inside you have your coacervate nicely sitting. So that's good. Now let's turn our attention to liposomes. So now if you encapsulate everything in liposomes and first only trigger SPS, and this I'm not showing you here, but we We prove this by also doing double emulsion experiments by using a slightly different trigger. So if you only induce SPS at first, something strange happens. Not only you get, of course, the dextran-rich phase formation, but you get this kind of small bursts at the membrane because dextran likes to wet the membrane. This is known. In a DexPEG system, the dex will always go to the membrane. So not only it goes to the membrane and form these bursts, but it also takes the polylysine along with it. Now, mind you that these are not coacervates. These are simply recruitment of polylysine at the membrane. And you can see a zoom in of one such bird where you can see that the lipid fluorescence and the corresponding polylysine and dex fluorescence coinciding. And these birds are still free to diffuse. Okay, so they can they follow the two-dimensional diffusion at the membrane. You can even see them fuse here together. So these are still 2D free in the membrane dimensions. Now, what if you trigger both SPS and APS? When we do that, I will play the video. Something even more interesting happens because now the dextran starts to wet the membrane. It even basically turns the liposome into a small flower. And it starts harboring these coacervates inside those condensates. Yeah. And you can also see from here that the dex the dex signal and basically the PEG signal co-localizes very well. In fact, the PEG signal is always within those dex regions. And if you do a 3D confocal of such structures, you get these beautiful flowers with many of these petals having coacervates. So here is one coacervate, here is another, and some are empty. And you can see this even better in cross-sectional views across the entire liposome. So you have to realize how complex this whole structure is, right? So there's all these petals are protruding all over the place, and many of them are actually harboring these our coacervates. And if you look at the diffusive behavior of these condensates inside those buds or these dex phases, they are quite different. So for the SMC buds, the mean square displacement actually follows a two-dimensional, a 2D diffusion, as you would expect. But for the mean square displacement of these of these condensates confined within these petals, they actually are restricted by the size, which becomes apparent at higher times. And what you get is kind of a choral diffusion, and the choral size is about a micrometer, which fits very well with this small condensate patches. So we have two completely different ways of confining coacervates basically within liposomes. So if I summarize this, what we have basically shown or is kind of a control that we can gain over condensates within liposome by using segregative phase separation. So if you don't want membrane interaction, you can simply get an APS within SPS, just basically sitting in your vesicle. But if you want membrane interactions, you can actually do two different things. You can either have your condensates within like big domains, or also basically sequester your components to some sort of SMC buds. So we think it's a very, very interesting way of organizing or complexifying your synthetic cells in future. And I think this could be interesting, such strategies. All right. And now I go to the third and the last part of the talk is, of course, I mean, we like microfluidics and all other techniques, but we are always thinking, okay, can we do something different? Can we make synthetic cells in yet another way? And this time we were thinking, can we spray synthetic cells? And when I say spray, I basically mean electrospray. So electrospray is a soft ionization technique, which is, I mean, now immensely useful with mass spectrometry, mass spectroscopy, sorry. And the basic principle is like this. If you subject a droplet to an electric field, it will elongate, because as the surface charges accumulate, the surface tension is opposing this increase in the surface area. And when the surface tension and the electric field, basically these two forces are in balance, what you get is kind of a conical shape of the droplet, which is known as the Taylor cone. And if you increase the electric field further, you basically start spraying the emulsions, or you basically start spraying that material in the form of droplets. So we thought, can we use such an electrospray technique? So we built one in-house, and it's a very simple setup. You basically have a ring electrode, and you have your cannula, and and then you just simply spray your droplets. And you can, so this is actually a video. You can see that Taylor cone, and it is in fact spraying some droplets, but it is nice and stable, and that's why you don't see it. But we did characterize how where we get this Taylor cone formation. So, for example, here you see an intermittent Taylor cone formation. But if you start increasing the voltage, you can get to the stable regime. But if you increase it too much, it becomes further unstable, and then it just simply has large droplets dropping basically. And now we spray our favorite system, the DEX in PEG droplets. You can see them here. We can spray them nicely. Sure, the size control is not as good as in microfluidics, but we still get a decent size distribution. But our idea here was actually, can we use this segregative phase separation plus SUVs, so small unilamellar vesicles, to achieve some sort of a electrospray induced reorganization at the boundary of our droplets. And for that, we used the following lipid composition to make our SUVs. So there is DOPC, DOPG, there is DOPS to induce fusion. And then there are these pegylated lipids, because remember, we have this DEX-PEG system. So we are thinking whether we can basically increase the interaction at the interface, and which we will soon see is very important. So this is how it looks like now. The experiment is like this. You are basically spraying dextran-rich phase with SUVs into a plate which has PEG in it, or the PEG-rich phase. Yeah. And what you get is quite something. You get these beautiful droplets, but surrounded not by a smooth layer or something, but basically a lipid nebula. Right. So the lipids are actually encapsulated within the dextran phase, but they really go to the interface and form these beautiful structures. And we can actually control how big these nebulae are based on the SUV concentration put in the DEX. So if you put less concentration, you start getting smoother assemblies, but also the time dictates the assembly. So we simply kept them for a few days, and over 3 days or something, they not only become kind of smoother, but also start to interact with each other to form this kind of a networked assemblies. You can see the lipids kind of touching each other and actually adhering to each other to form this kind of multicellular assemblies. And these are structures floating on top of this PEG phase. Now, a few negative controls. I like negative controls. They are very important and good to kind of decipher what's happening. So this is the positive control, where we simply electrospray in presence of the pegylated lipids. If you apply no voltage, of course, you simply get a blob with all these SUVs, as you can see here at the bottom of the dish. If there is no ATPS, again, that also does not work. So the SUVs really need something to build the interface upon. If you invert the system, so if you spray PEG in DEX, that also works. Perhaps not as good as DEX in PEG, but it still works. You still get this SUVs at the interface. But if you do not have pegylated lipids, that does not work, because as you can see, the SUVs remain sequestered inside the DEX droplets. So these pegylated lipids are really important for SUVs to go to the interface, to interact with the PEG phase, and while getting electrosprayed, also get scrambled and form this kind of this nebulae. Now, another interesting thing that we saw was when we kept on basically incubating these structures for a week here, what we got was quite striking. We got basically two different populations. So at the top of the dish, we got these floating assemblies. But at the bottom, what Pim observed was he got these vesicles, uh, individual vesicles. And if you look at them closely, they actually almost look like liposomes. So they have a very, very thin lipid shell. And actually, all the extra lipids, if you change the focus to the bottom, all the extra lipids have gone and accumulated to form a kind of an aggregated pocket. So these are really interesting structures that are happening over quite some time. And we wanted to characterize them a little bit more. So we made two different populations of SUVs. So one green and one red here. In case of floating networks, these populations do not really mix, saying that these are very static assemblies. They do not have a lot of reorganization within them. Also, if you do FRAP, it shows no recovery. But in case of sunken vesicles, you clearly see the overlap as a yellow color. And again, suggesting that these smooth lipid shells might be actually very close to lipid bilayers, but we have to do further studies on them now. So actually, that's the three stories I wanted to talk to you about. These all these three are published. So if you're interested, please have a look. The first story is about using ELPs and double emulsions as synthetic cell platform, then using segregative phase separation to further regulate the condensates within vesicles. And the third story is actually using a completely different technique of electrospray to make vesicles. And when I started making this presentation, I couldn't help but kind of look back at how it all started and how are things going. So I went into the synthetic cell business with Ola, with making these vesicles, and then we did some weird and crazy things like dividing liposomes purely mechanically, but also actually grow them using membrane tension. And then we also had a collaboration where we looked at where we use liposomes to look at drug screening on an on-chip platform. And at the end of my postdoc, I really became interested in those condensate membrane interactions, or basically condensates in vesicles, and that really sparked an entire interest in condensates. And then starting with condensate membrane interactions, but not just that, just simply using condensates as the containers itself. So we developed these actinosomes, which is actin-based containers, basically to produce, for example, proteins here. I also went into biosensing, so completely different topic. What you're looking at here are liquid crystals that are kind of sensing lipids here. So again, still kind of related. And then we went to the stimuli responsive ELPs, further into the further complex systems of APS plus SPS in liposomes. And the fascination for protein phase transitions continue. And I think all of this now can lead to even more exciting things of now we want to actually really go into biochemistry and tag different condensates with enzymes and bring about biochemical reactions. And the dream is to have different sorts of condensates within a vesicle, just like how a living cell does it, but on a much more remarkable level. So yeah, of course, I mean, this is thanks to a great team that I have, very enthusiastic people, including also master students. So Ketan, Chang, and Pim are the three guys who did a lot of took lead in the three stories that I showed. But also there are lots of students involved, and we have a new team who is also going to work and continue with the synthetic cells. So also thanks to all these collaborators and funding. And lastly, thanks to you for joining and paying attention. And I'm ready to take questions.
Thank you so much. This was a fascinating research talk, but I also really love the visuals.
Thanks.
There is a lot of questions in chat. Can you see chat?
Oh.
If you could read a question out loud, because people watching the recording don't see the chat.
Yes. Shall I stop sharing, or that's
yeah, maybe that's gonna make it easier.
Okay, good. So, oh, sorry. Where did it go? Sorry, Kate.
I can read the questions
No,
too if you
it's
want.
okay. It's okay. I got it. So what scale
Awesome.
can you produce in microfluidics? For example, if I wanted 1 ml of sample volume, would it be possible to create that much of liposomes in a typical microfluidic setup? Right. Nice question. So at least I will say for us, we are not using microfluidics to have such a kind of scale up. Especially with liposomes, having a 1 ml sample volume with, I guess, a high density of liposomes, I think that's difficult. But with double emulsions, it certainly is possible. So we typically produce double emulsions at, it's possible at 1000 hertz, for example. So we are making millions. Even at 100 hertz, you can make millions in an hour. So for this directed evolution experiment, we make a lot of these double emulsions because actually we do fluorescence assisted cell sorting. So I think it's possible with double emulsions. With liposomes, difficult. But I guess there are lots of other labs who are using such microfluidic tools to make also liposomes at a large scale, including using OLA. So yeah, I think that's possible at one point. So when you make synthetic cells with water in oil emulsion, how do you present the more hydrophobic components of the translation mix from partitioning into the oil instead of remaining in the aqueous phase with the rest of the reaction mix? Right. So I suppose you're talking about if you have, I don't know, maybe more hydrophobic proteins in the inner aqueous, how would you ensure that they remain in the inner phase and not go to the oil phase, right? Of course, I mean, yeah, that can be a problem. Normally, you want it depends upon what you want. I mean, we are also making use of this. So for example, we are using some sort of amphiphilic proteins or peptides to actually make them go to the interface. But really avoiding the water oil interface is difficult unless you really have a lot of surfactants to basically completely cover the water oil interface and have a strong competition with those kind of molecules. Yeah. But for example, I mean, we have encapsulated TXTL, and that works wonderfully. I mean, we can nicely produce proteins inside the cavity without any problem. Can you merge two microfluidically produced vesicles into one to create a larger compartment containing the components of both vesicles? That will require some sort of a fusion machinery, I suppose, right? I guess in principle it's possible, either using snare proteins. People are also using DNA-based fusion machineries. We have not done it. I think it's I don't know why would you want to do this. Another option is also actually encapsulating a complete liposome within another liposome during the production. I think that could also be possible if you want a liposome within a liposome. Or oh yeah, but yeah, you are talking about okay, you are talking about combining two liposomes together. Yeah, you can do this using fusogenic agents. If the pH of the continuous medium is changed, how does it affect the cargo of double emulsions? Wouldn't oil shell prevent the molecular transfer from outside to cargo? Yes. So the oil shell, of course, is a barrier, but you can still equilibrate the pH across the oil shell. That's what we see. It takes a little bit of time, but you certainly can do it. So it's not completely impermeable to such proton efflux. Okay, you can certainly do that. And of course, I mean, the pH will affect whatever is encapsulated inside, and that's how we trigger our ELPs, for example. Beautiful images. Thank you. In the experiments with inducible formation of coacervates, are the conditions compatible with cell-free translation? Yes, I would say so. So we have not exactly measured the pH inside, but I don't expect it to be super low as well. And I think we can also trigger at much higher pH. So as I said previously, we have in other experiments, we have definitely expressed proteins within those double emulsions. So that's certainly possible. Yeah. Have you thought of doing electron microscopy on the nebulae around the liposomes? Those are very interesting structures. Yes, Birol, I completely agree. This will be interesting to do. I have not. No, we have not done it yet. Should be should be possible. These are very stable structures as well. Will be good to see what exactly the structures look like, especially if they are continuous or still in the SUV state, so still enclosed. But just aggregated lipids. How stable are your double emulsions? Do they need agitation to prevent collapse? They are very stable. They are very stable. They are stable across temperature of at least from 4 degree to 50 degrees, pH from 1 to 13. Also, they are very stable against osmotic shocks. So they can increase at least, I think, 60% of their volume. And also they can shrinking is not a problem. I don't exactly understand. Do they need agitation to prevent collapse? So if you're asking if they aggregate, not really, because we make a batch and we keep it in the fridge, and we can also use it over weeks to simply pipe it up and down and take them out. They're very stable. I assume for double emulsions, you do not de-wet the droplets. Have you characterized the thickness of the oil between lipid layers? Yeah, thanks, Prathamesh. Yes, we do not de-wet the droplets. That's the whole idea here. Using fluorinated oil here. The thickness is about a micrometer. And I think you can increase or decrease it by further changing the oil, the pressure on the oil phase. But yeah, typically about a micrometer. Yes, I think.
I think you went. Yeah, I think you went through all of. Oh, next one.
Oh, there is one more. Inspiring for upcoming microfluidics. Perfect. Good. What kind of mass transfer do you see being possible between the coalesced and uncoalesced drops over what type of time regime? Sorry, I'm not exactly getting this.
May I explain?
Yes, please. Yes.
So you said that over time the drops were coalescing and being bigger drops.
Right.
So my question is, if we want mass transfer between these droplets, over what time length could we what kind of mass transfer do you see happening? Because I'm assuming that in shorter times maybe there's different types of mass transfer because of the shell that's covering the droplets.
I guess you are talking about the coacervates that are within the vesicles, right?
Yes.
Yeah. So I mean, two things are happening there. First is they are literally physically fusing with each other, right? So that's one thing, and I think that's the dominant process here. And the other thing that can happen is Oswald ripening, and then that's why the mass transfer. But yeah, I mean, we typically see within minutes that you will basically end up with a single coacervate. So it depends on the how confined your vesicle is, how small the vesicle is, and how many nucleation events are forming, and the diffusion. Of course, you can also think about not making them fuse, and we are thinking in this direction as well. How can you make them individual entities and so on? I hope that answers the question.
Yeah. Thank you yeah.
Thanks.
Thank you.
Thank you so much. I don't have any other of my own questions. I was really intrigued by the stability. It sounds like you're saying they're really rock stable. That's amazing.
Yeah, the double emulsions really are. Yeah. And I think that's why they can be interesting, for playing around as confinements, basically.
Yeah. Because we've been having our own problems with just like normal, not fancy emulsions, just emulsions that we do for encapsulation of aqueous solutions, just the single emulsions. And we've been having problems with those kind of a collapsing over time.
Okay. Check these out.
Yeah, we definitely will.
Yeah, nice. Good.
All right. I think if there are no more questions, thank you so much, Siddharth. Thanks again for an amazing talk.
Thanks a lot, Kate.
And congratulations. I can appreciate how
Oh,
difficult those experiments are.
Thanks.
And thanks, everyone, and have a great rest of your week.
Yeah, you too, guys.
Bye.